So this is our chapter 2. We're working on chapter 2.1 to start out. If we were in class, we would do this introduction activity, but I'll go over it and you can sort of look at it on yourself. It says a combination of different physical characteristics make each person unique. Many physical differences, such as the color of eyes, hair, and skin, are due to slight differences in the genetic information that each individual received from his or her biological parents. One characteristic determined by your genetic information is the ability to roll your tongue. So this is one of the many physical characteristics that we have that sort of is a genetic thing from your parents. It says, some of our discussions, it says, were you able to roll your tongue? So for me, I can roll my tongue. I think my parents can too, but I haven't checked. At least one of them should have. And then we look at it through the class, so we can't really do that. Some different things. This is all about rolling your tongue. But if you look on the right-hand side here, some of the other characteristics, I know feet are gross to me, but some people have a longer second toe than any of their other toes, so that's also a genetic thing. People have their thumbs could be straight when they hold them, or they can curve quite a bit, like you see in this picture. So checking out your thumbs. You could have hair on your fingers, basically, so that hair that grows between your on your knuckles, I guess is what it is. That's a genetic thing. And then your ear. If you look in this picture, if I can do that. This part of this is a really blurry, but this part of your ear, so your earlobe, it's a genetic thing that your earlobe could be attached or detached. This person's looks detached, so this part of their ear is not attached to their head, it's actually off of their head. But that's a genetic thing, so being detached or attached. For me, my ears are attached, but many people's are detached. So that's another genetic marker. So what is genetics? Well, it's the scientific study of how genes work to determine characteristics and to resolve how genetic information gets passed on from parent to offspring. So it's a study of how our genes work, and our genes are in our DNA, which we'll get to in a second. Chromosomes, which are located in each cell's nucleus inside of our bodies. There is chromosomes, and they are made up of strands of DNA. DNA is deoxyribonucleic acid that contains the instructions for making proteins. Humans have 23 pairs of chromosomes, so that's 46 chromosomes in total. And 23 of those come from your dad, and 23 come from your mom, okay? So you're made up of half of your mom and half of your dad. It's not that simple that it's like 1X comes from your mom, 1X comes from your dad, or chromosome, sorry. But you are getting pieces of information from both your mom and your dad to make up those 46 chromosomes, okay? A gene is a section of a chromosome that carries the instructions for a specific protein, and these specific proteins in our body, they code for certain things. So proteins, they code for different traits, I guess you could say. And scientists have done a lot of research into this, and they've actually mapped the human gene. Obviously, there's lots more to be found out, but they can link certain genes to certain traits, which some people might think is a little crazy, but it is something that can be done, and mostly this is done not in the sense of trying to, genetically engineer people, but it's used in a sense to be able to figure out diseases. So you could screen yourself to see if you are possibly going to have a certain disorder. Something runs in your family, they can check to see if you have that gene. So that's something that gene mapping is definitely something that's used for the good. So what is DNA? Well, it's deoxyribonucleic acid. That's what DNA stands for. If you look at the word DNA. And it's a twisted ladder shape, a double helix that contains genetic information of the cell. This is made up of chromosomes, which are made up of genes. So it's like DNA is inside of chromosomes, and so are genes. So the whole chromosome is DNA. Inside the chromosome is our genes. So they're all connected in a way. This says it's a double ladder-shaped helix. And maybe I will just show you a picture of what that might look like. When we think of a double helix, I always think of like a corkscrew or like just looking at a screw, but this is definitely the picture of what DNA would look like. So you see that it's got two sides, that's why they call it a double helix. And each side has certain pieces connected to it, basically. So it says a chromosome is like a cookbook. It has a whole bunch of instructions for making different things, and a gene is like one specific recipe, and it's instructions for making a specific thing. Okay, so chromosomes, the whole cookbook, a gene, just a specific recipe. Okay, so that's a good analogy, I think, to realize that genes are really specific, and chromosomes have lots of genes inside of them, just like a cookbook has lots of recipes. A karyotype, so this is another word. It's chromosomes are investigated using a karyotype. They may be used when diagnosing genetic disorders, and they may be performed on a child, an adult, or an unborn fetus. This unborn fetus one is amniocentesis. So you may have heard of that before, but when you're pregnant, you can get this genetic screening where they will check to see how your baby is, basically, see how they are growing, and see if there's any sort of problems genetically. In the nucleus, the chromosomes are not neatly organized. So when we look inside of a chromosome, they are not all in a row. They're just in a huge pile of spaghetti, or that's what it looks like. A karyotype is an image of all the chromosomes in one nucleus that have been matched up into pairs. So if you were to look at a chromosome or a nucleus, inside of the nucleus, that's a nucleus, you might just see a bunch of squiggly lines, and you don't really know. Well, scientists will take those chromosomes and they will actually, sort of like matching. They'll match up the ones that are the same, and then they'll sort of line them up. So you'll get sort of a picture of what chromosomes might look like, okay? And they'll match them up so that in a human there's 23. Okay? So you got 46 chromosomes, and they're in groups of 2 to make your 23 pairs. They're always measured first, arranged from smallest to largest in length. And the longest or largest ones would be labeled 1. So these would be our biggest ones, and then as we go down, our smaller. Again, is it going to show you? I will show you a picture of that as well. So if we go back over to here, if I show you a karyotype, you can see what a type of karyotype might look like. So this would be considered a human karyotype, so they are putting these chromosomes together, matching them to the ones that look alike, the biggest one at the top, all the way to the smallest at the bottom. Okay, so that is what a karyotype would look like. But when it's in a cell, it's just going to be a big jumbled mess, basically. And that's what scientists do to be able to sort it out. Okay. The last two chromosomes in a karyotype are the sex chromosomes. Okay, so the very last ones, if you look back to that picture that I have here, you see that at the very end we've got 22 and then we've just got X and Y. So the 23rd pair is the one that determines whether you are male or female. And that is known as our sex chromosomes. These determine an organism's gender. I wouldn't say gender, I would say sex. So this is something that we will fix here. So your biological sex. Yeah. I can't move this up. Oh, there we go. So your biological sex of an individual has two X chromosomes. The sex is female. And if you have an X and a Y chromosome, then your sex will be male. The Y chromosome is always considerably smaller than the X. So it's always a challenge to match. So this Y, see how small it is compared to that X? It's the one that we can't really match up. Oh, so it shows you one here. So the one that I was showing you on the Google Images was a male because it had an X and a Y, and this one is two X's at the end, which means that they would be female. There's a human karyotyping activity that I normally get you to do. I'm not going to get you to do it, but it is something that's sort of cool, I guess. So we'll skip over that. Maybe I will post it on the Google Classroom, something that you could do. I don't know if it's actually it might be in your booklet. So if it is, I'll double check that and post it on the Google Classroom for you. There are over 2,500 genes that are located on our 46 chromosomes. This means there are over 25,000 proteins created in our bodies. This is interesting. If you would take all of the water out of our bodies, almost 50% of the dry mass left behind would be proteins. So 50% of that dry part of our body, the part that's not water, is protein. So we're always creating proteins in our body to do certain things. Some different types of proteins, just to show you. An enzyme. We know an enzyme is something that will speed up a reaction or break something down. An example is amylase, which is in our saliva. That's something that will help to break down things. You've probably put food in your mouth before and let it disintegrate, and then eventually it will become just mush, and that's because the enzymes are breaking that down. Structural types of proteins. These are proteins that will attach to other proteins to be supporting. An example of that is keratin and collagen. Keratin is in our hair and our nails, and collagen is something that is helping to keep our skin together. So, as you get older, your skin gets more saggy. Well, that collagen is really important to keep that the support inside your skin. Transporting, so there's lots of proteins that help to transport materials around your body. One of a good transporter is, well, it doesn't give you an example per se here. It says cell membrane helps to pump stuff in and out. So in the cell membrane, right in the wall of the cell membrane, there are different proteins, and there are things that will help move things in and out of the cell. We also got hemoglobin, I think is a good one, which we just talked about in the last chapter. Hemoglobin is a protein that's found on your red blood cell, and that's something that helps to move around your iron. And then the last one here, hormones that act as signals to coordinate and regulate activities in the body. A hormone that is an example of this, that's a protein, is insulin. This is released from your pancreas, and that's used to regulate your blood sugar levels. So when your blood sugar levels are high, it'll release insulin to bring it back down. So is this making more chromosomes? So how do we make more? Well, in our body every day we are losing chromosomes, or we're losing cells, they're dying. And we're using this to make new ones. So on mitosis is on the, I don't know if this is gonna give you much on it. Okay. I'll go over this one really briefly and then I will maybe stop right there for today. But mitosis is when we replicate our cells. So if we had a skin cell and it died, we have skin cells dying all the time. That's why you have that sort of skin will be flaking off of you. That will need to be replaced. And what replaces that is mitosis. So one cell at the very first here, your parent cell is what we call it, will replicate. So it'll create a copy of itself so that at the end we've got two copies. We've got the parent and then we've got an exact copy, okay? So that's what mitosis is. It creates a copy of parent cell. Meiosis is a little bit different, and this happens in your sex cells only. So this is something that it doesn't want to create a full copy, it wants to create a half copy, I guess is the word. So we start out with a parent cell at the first. Our DNA will replicate, which will create basically two copies. And those copies will then divide again, so that we're left with 4 cells at the end, and they each have like half of the original, okay? So we started out with an original, and at the end we have 4 cells that are half the genetic material of the original. Okay? So that is mitosis and meiosis, and to be honest, we're not going to go into it a whole lot deeper right now, and possibly won't at all in this class, just because of time's sake. So there's some vocabulary that goes along with mitosis and meiosis. To be honest, I'm just going to, let's skip over that part for now, because I just want to make sure we have enough time to do everything we need to in this class, and I'm worried we won't. So we're going to skip over that part. But some ways to remember mitosis and meiosis. Mitosis is remembered as mitosis. It produces 2 cells, 2 copies of the chromosomes. And mitosis is known as a diploid. This is another word you need to remember. So di means 2. It's a diploid cell. And basically it means at the end we have 2N. And N means the original. So we've got 2 N's, we've got 2 of the original. Meiosis, on the other hand, is remembered as meiosis. I don't know if I ever use it that way, but it produces something called haploid cells. And hap, it means half. So they're half cells. So at the very end, they create something that are haploid cells. They call that 1N. And this is one copy of each of the chromosomes. from the original being split into 4 different cells, okay? So this is sort of how, well, this is exactly how our body creates sperm and eggs. Because imagine if you took a body cell from your parent and a body cell from your other parent. If we were to put those together, then you would have too many chromosomes. You would have, I could just show you a picture of this. So let's say this is mom and this is dad. And let's say I don't have enough room to make enough chromosomes, but let's say they each had 4. Well, if they put them together, they would create an offspring that had 8. And everyone in your species should have the same chromosomes as you. So these are one species, and now they're creating a different species. That's not possible. So what actually happens instead, that's not possible. What happens instead is we have mom. I'm going to put 4 again. And then we have you. So you have 4. But what actually happened was that your parent produced 2 cells. And this parent produced 2 cells. And then these 2 cells went together to make you. Okay? So inside of their body, they split their DNA into 2 separate cells, and then they were mixed together to form you. Okay? So that's what meiosis is. Meiosis, whatever you want to call it. All right? And that's what's happening in your body or to create you. All right. I only have a couple slides left, so I may as well finish those off. So fertilization, it says when chromosomes in a male gamete, and a gamete is the sperm for a male, and a female gamete, which is the ovum, or the egg, as we just call it, egg and sperm. When they join together, they make a diploid cell. This cell will undergo mitosis to produce a new individual with combinations of characteristics from your mom and your dad. Sexual reproduction results in almost limitless combinations of genetic material from 2 parents. Crossing over serves to increase the variation of the genetic material. I don't know if they have a picture of crossing over. They possibly do. You know, my favorite people who show crossing over are the amoeba sisters. I don't know if I'll be able to get a picture of it. I will definitely put, I'll put their video on the Google Classroom for you. Yes. This picture will show you everything you need to know. So the amoeba sisters, they are really good at this. So what they show you is you don't necessarily, let's say your mom's was the red chromosomes and your dad was the blue-purple chromosomes there. So you don't necessarily just get one red chromosome and one blue or purple chromosome. You get a mixture. So there's a thing that happens during this process called crossing over. And basically little pieces of each chromosome will go back together. They'll make a new puzzle and glue themselves together to make like a whole new person. That's why like every child that your parents have it's not the exact same. They're not copies of each other because there's limitless possibilities of genetic material being reorganized due to crossing over. Okay. So they do show you a picture here. So yeah, they've got on the top here the female. So the female her egg goes into meiosis. It splits into one in each of those. the male's sperm goes into meiosis and splits into 4 pieces. And then if we put all of those pieces together, those are all of the possibilities that we could have as an individual. So many different possibilities, and think about it, there's many, many more. We have more than 2 chromosomes, like it's showing here. We have 23. So if you think about 23 and then all of the possibilities that could come out of that, you would have quite a variety. So every time someone has a baby, they're not going to be a duplicate of the old baby, no, but the other offspring or child. Okay, so there's some questions that go along with this on page 92. I just want you to do question 1 to 5. All right, let me know if you have any questions about any of this, and I will try my best to help you out.